Thank you very much, Tamara, for agreeing to give a Build a Cell seminar, and you can get started.
My pleasure. Thank you for inviting. It's an honor to be here today and present to you and your community of Build a Cell a little bit about what my lab is doing, both in the past and more recently. So the title of this talk is All Atom and Coarse-Grained Simulations of Complex Biological Systems. And I will focus on the research side on how we model proteins, and in particular, adhesion proteins, those proteins that are involved in creating physical connections between the internal part of the cell and the cytoskeleton, in particular, with the outside of the cell. I'll start with introducing myself, who I am, where I studied, and what is my background. And then I'll very proudly introduce you the member of my labs that are making the current research possible. I'll show you a little bit of what were the past projects during my postdoc and also at the beginning of my lab, and what are the ongoing projects that involve all of the new postdoc and students that are here at the University of Utah. And then I'll focus on coarse-grained and all-atom simulations of adhesion mechanosensing. My background is in mechanical and biomedical engineering, so I've always been interested into the effect of mechanical forces on proteins and macromolecular complexes. And I ended up studying in much details how integrin-based adhesions respond to force in the sense of how they assemble into macromolecular complexes and give rise to nascent and focal adhesions, as well as how they actually change conformation in response to stresses and deformations, in particular from the outside, from the external environment, which mimics difference in tissues, for example, as well as when we think about metastatic cell migrations, we are thinking about cells that come across very different tissues. And one of the properties of those tissues that vary considerably is the mechanical properties and the stress, the deformations that they can exert on cells. So the idea of studying how adhesions respond to forces have implications both in fundamental research and understanding how those proteins deform under stress, as well as how they assemble. So they change their kinetics for binding and unbinding under those forces, but also has implications for the study of pathologies like metastatic progression, for example. I come from Italy. I studied in the very first public technical university in Italy, which was founded in 1959. It's called the Polytechnic University of Turin. Turin is in the northwest of Italy, about a bit less than 2 hours from the border with France. This is an example, this is a picture actually of the Valentine Castle in the Valentine Park, where there is the architecture classrooms. This Polytechnic school offer courses in engineering, architecture, urban planning, and industrial design. I followed mechanical and biomedical engineering, and I received my PhD in 2013 with a thesis on the development of computational models of soft matter, meaning cytoskeleton and cytoskeletal assembly. Then I did my first postdoc at Lehigh University in Pennsylvania, in Bethlehem. I was in the Department of Physics, where I developed Brownian dynamics-based models of actin filaments reorganization in space and time, depending upon the concentration of motors and cross-linkers in the space. And we could capture different type of assemblies depending on those molecules present in the system. Then I focus more and more into the idea of building multi-scale models, not only characterizing the proteins as polymers or rods in space, but also accounting for their molecular structures. So what are the constituent atoms, how they interact, what are the weak interactions and the covalent bonds between them, and how those molecular details can be used to inform larger scale models that have lower resolution. So at the University of Chicago, I started developing those coarse-grained approaches, and using it, I became more familiar with statistical mechanics and the coarse-graining field for biological systems. I started my independent lab at the University of Utah in 2019. I'm part of the Biomedical Engineering department, as well as the Scientific Computing and Imaging Institute. I am in this building, which is the Warnock Engineering Building, and on this floor here, this is where I am currently giving this presentation. And over the course of now 5 years, I started my lab. I have currently 3 PhD students, Remy, Onkar, and Reza, 1 undergrad and 2 postdocs. There are more people that are not in these pictures. We are very active and keep expanding. Garima, for example, is a new master student, and there is a rotating student, Kadus, and a software engineer, Keith, currently helping with the projects. What is our mission? So we want to understand the molecular basis and the fundamental mechanism of complex biological systems. We have a project not only on adhesions, but also on microtubules. So we span between adhesion properties and adhesion dynamics and kinetics to the properties of large polymers and polymer networks, actin filaments and microtubules more recently. We use computational approaches, and we actually cover a range of scales. So we still run a lot of atomistic simulations, both equilibrium and enhanced. Enhanced meaning that we provide energy into those atomistic systems in order to increase the sampling and increase the amount of possibilities that those proteins can actually span configuration and span modes of existing. And we also developed an Brownian dynamics model based on particles moving in space and moving in this very chaotic environment, which is agitated by thermal effects. We introduce in those Brownian dynamics models kinetics and forces when we want to study, for example, the effect of external forces on those agglomerates or assemblies of macromolecular components. The cells, as you guys know in this group, are special in the sense they don't have any analog in the physical world, in the world that we see around us. And one way that we can emphasize their difference with the physical world is by thinking of thermal energy and its effects on the dynamics of macromolecules and smaller molecules in the space of a cell. There is molecular crowding. Molecular crowding is that effect such that those components basically are present in high concentration, and the high concentration of macromolecules actually reduces the volume of solvent that is accessible to other proteins and macromolecules in the space. This has the result of actually increasing their effective concentration. And then there is another important feature of the cytoplasm, the environment inside cells, which makes it unique with respect to the physical world and more like macroscopic objects, which is the fact that it contains an abundance of mechanoenzymes. Mechanoenzymes convert the chemical energy into mechanical work, and this is an example of myosin mini filaments sliding 2 antiparallel actin filaments in opposite direction in order to generate local stresses and cell contraction. So the fact that there are mechanoenzymes means that there is an external energy source in addition to the Brownian effects from the thermal forces. This brings this system out of thermodynamic equilibrium and makes the cytoplasm a very unique material with no analogs in the physical world that we see around us. So this is a little bit of a summary of computational models of the cytoskeleton that I've been developing as a postdoc and now more recently in the last 2-3 years with my research group. So this movie here is the movie of a simulation of cytokinetic ring assembly in fission yeast. You can notice how those semi-flexible polymers that form by elongating in space, in the space of a simulated fission yeast cell, actually reorganize from a very randomized network and disordered assembly of filaments into a more organized ring that will contract to help with cell division. This is a representation of a budding yeast cell where we simulated the elongation of cables. And then I had other models of cytoskeleton contraction and reorganization. In particular, we were studying how different concentration of motors and cross-linking proteins could change the reorganization of this network, again, from disordered components and filaments to more ordered assemblies, including bundles and kind of stars. More recently, with my group, we have simulated the assembly of bundles of actin filaments cross-linked by fascins. And we were particularly interested in understanding how different binding modes of this cross-linking protein could give rise to different type of hexagonal bundles of filaments, in the sense that some of those fascins could cross-link the filaments with higher or lower energies. And with our algorithm, we could capture some of the relative differences in rotation of the single filaments that we then compare with cryo-EM microscopic images from the Alushin lab at Rockefeller. We are simulating microtubules now, and we are studying the very classical question of what is GTP hydrolysis doing in terms of destabilizing the lattice of a microtubules. Those are examples of bottom-up type of models where we take atomistic trajectories of GTP-bound versus GDP-bound lattices. And we study basically the relaxation of those protofilaments when they start bending outwards to give rise to those flared tip morphologies. And we are comparing and contrasting the effect of nucleotide hydrolysis on those flared tip morphologies. And those are the projects more recent about adhesion mechanosensing. We look at single proteins as point particles on 2D surfaces, mimicking basically their diffusion and their assembly during adhesion formation. This is an example of a whole-atom MD simulation on an integrin intermediate, where we can see that there is this transmembrane protein that has a very large extracellular part, followed by 2 transmembrane helices and very short cytoplasmic tails that basically fluctuates under thermal motion at equilibrium. So we are taking those single protein trajectories and try to understand the mechanism of activation, which we will see in this specific presentation. And then I will go over this other model of adhesion assembly at the lamellipodium, where we have integrin receptors here binding external ligands, coupling with the actin filaments, and exerting and transmitting forces both inside out and outside in. This is an image of a more detailed model where we are studying protein-protein interactions, how basically at the level of the interface between integrins and a receptor containing the arginine, glycine, and aspartic acid, RGD motif. We are studying exactly the molecular details of these protein receptor interaction and how individual secondary structure elements at the ligand binding site move because of the formation of weak interactions between the RGD motif and the ligand binding site. So let's start with an experimental image. Those are 3T3 fibroblasts from the Oaks group at Loyola University at Chicago, showing us that cells plated on soft substrates are actually round, whereas as soon as you plate them on stiffer substrates, they become more spread and they present a lot of bundles of actin filaments, which we call stress fibers. So cell spreading is actually a mechanism that is sensitive to substrate rigidity, so that we can capture those two types of phases of spreading just by varying the stiffness of a substrate. And what do adhesions do? This is another immunofluorescence image where we can see both paxillin staining, which is a protein of adhesions, and actin staining, and both. The question is, what happens with the adhesions? So if we look on soft substrates, the number of adhesions is much lower than on stiffer substrates. So not only the cells tends to increase more on stiffer matrices, but also the number of adhesions increases. So adhesions respond to the external stiffness, and that's something that we have been modeling for quite a few years now. Our general viewpoint to understand how adhesions respond to the rigidity of a substrate is basically by thinking of the leading edge of a cell as the group of three main molecular machines. The first molecular machine is the one provided by the actin filaments polymerization and pushing against the leading edge, where they establish that net force, which provides membrane stretching and membrane tension and pushing of the membrane forward. Those actin filaments don't operate in isolation. They are connected to a second molecular machine, which is provided by the molecular motors. Non-muscle myosin 2A and 2B, for example, localize a little farther from the edge membrane and they contract. So we have those two opposing forces, the pushing from the actin filaments polymerization and branching at the very leading edge, and the myosin motors contracting this network at, farther from the very leading edge in the lamella region. The third machine that is the one we are mostly interested in is provided by the adhesion complexes. So this is an example of how adhesions are formed or how they look like if we look at them from the vertical direction. We have those integrin transmembrane proteins that create the direct contact with the substrate. They have this ligand binding site. They can bind, depending on the integrin isoform, different type of ligands like RGD containing proteins or also collagen and other type of ligands. Then there is another layer here, which is the mechanosensitive layer given by tallinn and vinculin. Tallinn and vinculin directly respond to force by either stretching and elongating or accumulating at the site of adhesions assembly. And then those two layers connect with the actin filaments and the cytoskeletal proteins. So the main question that we wanted to address with our modeling work was how adhesions respond to the external stiffness of the extracellular environments. And we looked at these from two main temporal and spatial scales. The larger is the one that we could capture with Brownian dynamics. So using models that simulate point particles as individual integrin adhesions and capture the dynamic of assembly and disassembly into those larger complexes. And the second length scale is the all atom scale, where we simulate individual integrin proteins and how they change shape under force, in particular, to try to see if they change shape basically more or less easily under the same amount of force, as well as what are the residues that that mainly provide the configuration to enhance this transition from one state to another state. So this is again the schematics of the cell edge. You can see here the actin filaments arranged in these double helix that assemble monomer subunits at their barbed end. This developed the pushing force to push the membrane edge forward, and is actually contrasted in the opposite direction by the action of the myosin motors that pull backwards those filaments. By creating their connection through tallin and vinculin to the integrin receptors that are here shown as spanning the plasma membrane and creating bonds with external ligands. So by creating those connections, these sum of forces, the pushing and the pulling in opposite direction, is transmitted directly to the extracellular matrix. And traction force can be measured from those substrates. Vice versa, if we change the rigidity of the substrate, this rigidity is sensed by the integrin receptors and transmitted back to the cytoskeleton in this inside-out versus outside-in signaling. How do we model such a complex system? So one of the main models that have been proposed in this field is called motor clutch model that considers individual integrins as clutches. They exist in two states, unbound or bound to the actin filaments. Those two states are regulated by first order reactions with a certain rate, K on. And in particular, the clutch can go from unloaded and unbound to bound in state 1 here and loaded in state 2. We can see here that this spring, which mimics the rigidity of the receptor, is actually stretched because once the clutch becomes connected with the actin filaments, it is subjected to this retrograde flow given by the action of the motors that are pulling in the filaments away from the edge, and by the continuous polymerization of monomers at the barbed end. Upon pulling these clutches, the K-off can lead to unbinding and relaxation of the clutch in state 3. K-off is a function of the force on the clutch, and we will see how what is called the catch bond kinetics actually directly regulate variation in off rate as a function of the force exerted on the clutch itself. So this is an example of a system that we simulated where we have basically a 3D system with two surfaces. The top one is the cell membrane and the bottom one is the substrate with an abundance of protein clutches that can bind and unbind the integrins here on the simulated plasma membrane. So those particles represent those integrin proteins. They are actually subjected to thermal effect. They undergo diffusion. To the diffusion, this has been measured with some single protein resolution approaches. And basically knowing the diffusion constant of those proteins, which is here of the order of 0.28 micrometer square per second, we could calculate the drag exerted on them during their motion using Einstein relation and simulate their dynamic in space. As they come close to a ligand, those springs represent both the ligand and the substrate stiffness. They can become connected to those springs, and upon a force which mimics the velocity of the actin filaments in one direction, they can build tension on those bonds. And the K-off here is the inverse of the lifetime of the bond. This type of simulations are implementing what is called overdamped Langevin equation of motion, where basically we calculate the displacement of those particles in time, knowing the sum of deterministic and stochastic forces acting on them and the friction of the environment. So by just simply summing the force contribution and dividing this by the friction, we can calculate the displacement at each time step of the simulation. So each integrin here is a point particle. They have identity I. If we monitor the deterministic and stochastic forces at each time step of the simulation, we simply sum them divided by the friction and multiply by the time step. So we can calculate with an algorithm called Euler implementation scheme, we can calculate the displacement at each time step of the simulation and then put all of our frames of this system in sequence and create those type of movies where we see continuous displacement at every time step and we capture the whole motion of the system. In this movie, you can also see how integrins transition from inactive to active and from active to inactive, because those two states are represented by different colors. How do they transition across those states? They basically can unbind ligands and become unbound and able to diffuse again in the membrane. The unbinding rate is calculated by this characteristic lifetime versus force relation, which happen to be a catch bond. Is a bond that basically strengthens under force. So you can think of this as the duration of the bond. And on this axis, on the x-axis, you are basically pulling on this bond with higher and higher force. So this relation between lifetime and force is telling us that the more we pull on the bond, the higher the duration of the bond is, up to a certain point, which corresponds approximately to 30 piconewton force. After this point, the lifetime drops into the slip phase of the catch-slip bond. So you can see here two relations. One is the characteristic relation of wild-type integrin, and the second in red is the relation of manganese-treated integrins. Manganese is able to both activate integrin with a faster rate and also to increase the lifetime of the bound state of integrin. So in this model of particles diffusing and binding and unbinding ligands, we basically modulated the unbinding rate based on those lifetimes and changed the spring constant of those springs on the substrate in order to mimic substrates with different rigidities, with low spring constants, representing softer substrates and higher spring constants representing stiffer substrates. So if we measure the amount of bound integrins at steady state, when steady state is reached in the system, which means that some of the properties basically don't vary anymore in time. In this case, it occurred after about 2-3 minutes of simulation. So if we monitor how many integrins are bound, we could capture this increase of bound integrins both with high stiffnesses or using the manganese conditions. Being the manganese condition, simply the second catch bond type, the one with higher unloaded affinity or lifetime at zero force, and higher peak in the lifetime at about 30 piconewton force. So by simply modulating this parameter, the manganese, the catch bond, whether it was representing manganese or wild type conditions, we could see the shift in the response to substrate rigidity. And if we simply increase substrate stiffness, we capture the increase of assembled or ligated integrins as a function of the rigidity itself. Another type of experiment, in this case, was based on traction force microscopy, provided us some evidence that not only the change in the catch bond changes the response of the system to substrate rigidity, but also changing the activation rate of integrin could basically shift the cells to spread more on softer substrates. So if we look at those images, we can notice here at about 3.1 kilopascal Young modulus of those polyacrylamide gels that the amount of spreading is enhanced when we use this activating mutant, S243E, and also the traction force exerted on the substrate seems to be increased. So in other words, not only when we modulate the duration of the ligated state of integrin, we can capture differences on soft substrates, like here in the sense of more adhesions formed, but also when activation of integrin is promoted, we can basically change the response of those cells on softer substrates. And this was quantified by Tristan Driscoll, who is now at Florida State University, and Martin Schwartz at Yale. This was quantified in terms of cell spread area and traction force. And what we should notice here is that on soft substrates, there is a shift in the response. So on soft substrates, when they use the mutant, they could capture enhanced area. And similarly, in terms of traction force, when they use the mutant integrin, they capture an increase in traction force, which is actually comparable to the traction force on stiffer substrates. So integrin activation, in addition to its bond duration, is another of those mechanisms that can shift the response of those cells on substrates with different rigidities, basically making cells on soft substrates responding as if they were on stiffer matrices. What is integrin activation? So we should look at the all-atom structure of integrin. Here is an example of inactive integrin, where you can notice the headpiece, which is the part on the external part of the cell. So this part here, this headpiece is completely bent. There are those two colors here, the blue and the red, representing the two chains or subunits of integrin. Integrin is a dimer, so two subunits that are non-covalently bonded for a total of about 2,000 residues, a bit less than 2,000 residues. The alpha chain here is in red and the beta chain is in blue. The ligand binding site is here at the interface of those two chains in this part here. And you can see that this ligand binding site is facing the plasma membrane in this conformation, meaning when integrin is bent and inactive, doesn't have a high affinity for binding ligands, and it appears that the binding site is not exposed towards the ligands directly. When integrin activates, it completely changes conformation up into an open and extended state. So you can appreciate here that the two chains in blue and red are completely separated laterally. The only part that they preserved as interacting is this ligand binding site at the top between the beta proper layer and the beta I domain. So the ligand binding site is exactly at this interface. When integrin binds a ligand, it starts binding in the beta I domain, in the beta subunit. And as the activation progresses, the ligand shifts towards the beta proper layer domain and ends bound to both. Those other domains that you see, the hybrid psi, EGF domain, and beta TD, are the domains of the alpha chain or of the beta chain, whereas the beta proper layer tie and the calf domains are those of the alpha chain. Beta chain here is in red, alpha chain in blue. How does this transition happen? So this transition is important for sensing of external stiffnesses and response to force. And so in order to understand how they modulate the response to force, we want to understand how it occurs. In other words, how integrins goes from bent to open. So in the field of integrin activation and dynamics, there are two main conceptual models that have been proposed. The first one is called deadbolt, and it basically says that the headpiece extends first by disrupting some of those interactions between the hybrid and the beta TD domain. And then second, the legs separate to separate the two chains. Whereas according to the second model, the switchblade, basically the legs separate first, and second, the headpiece extends. In other words, there is a little bit more coordination as soon as the legs start to separate, the angles at the knees of integrin here, the GNU and the beta knees, starts increasing magnitude up to the extended and open state. So basically, those two main conceptual models are different in terms of the order of events. So in the first case, the headpiece extends first, and then the legs separate. In the second case, in the switchblade, the legs start separating right away, and the headpiece is coordinated to this motion and extend completely as the legs separate in space of about 7 to 10 nanometer distance for complete activation. So we have some cryo-EM structures of integrin, and in particular of these beta-3 integrin, alpha-2b beta-3 and alpha-v beta-3, that have captured the integrin conformation in some intermediates that present the extended headpiece and the closed legs. So some intermediates that seem to be more close to the deadbolt pathway. So we decided to exert force on the headpiece to mimic ligand binding and substrate stiffness resisting the deformation. And we basically exerted forces of the order of 40 to 60 piconewton, which are pretty close to the experimental detected values, which go from few piconewton up to tens of piconewton, depending upon the technique that was used. And we monitored the degree of extension of the headpiece. So how fast is basically extending under force. In particular, we were interested in those beta-3 integrins, as I say, alpha-2b and alpha-v beta-3, the first one being highly expressed in platelets and implicated in thrombus formation and hemostasis. And the second ones, alpha-v beta-3, are important for solid tissues, fibroblast and other type of more solid tissues. Simulations of the extension here basically allowed us to capture this type of dynamics where the knees start extending and the headpiece elongating in the vertical direction. So when we monitored the extension of those two isoforms, we could clearly see that actually under the same amount of time and the same constant force magnitude, alpha-2b beta-3 presented significantly increased extension. This is normalized extension, so 0 represents the distance at the first time frame, and then how much relative to that distance the separation of the pooling group at the ligand binding site and the center of mass of the transmembrane helices varies over 20 nanoseconds of simulation. And you can see even here from those all atom reconstructions of those systems that alpha-v beta-3 seems to be a little more bent here at the end of the simulation with respect to alpha-2b beta-3. And it is more bent of something of the order of a few nanometers. So this was basically showing us that the ability to activate for those two integrins was different in terms of time for extending. It seems like alpha-2b beta-3 extends much faster than alpha-v beta-3. So we asked to Yunfeng Chen at the University of Texas Medical Branch if we could use this biomembrane force probe in order to pull on individual integrin proteins. And he pulled on alpha-2b and alpha-v beta-3, measuring both how long it was going to take for the two integrins to start disunbending at different force magnitudes from a few piconewton up to 30 piconewton. And then he also recorded once the integrin started the unbending, how long it took to unbend completely. In both experiments, the response was much faster for alpha-2b beta-3. In other words, alpha-v beta-3 integrins, both from endothelial cells and also from platelets themselves, took significantly longer to unbend and significantly longer to start the unbending versus to completely unbend. So the time to unbend was higher, as well as the unbending time. So in other words, if we think again about our results here, where alpha-2b beta-3 has a higher propensity to extend, this actually confirms the higher propensity for alpha-2b beta-3 to become completely unbent, both in the sense of taking less time to unbend, as well as unbending much faster. We said before that the activation of integrin is also mechanosensitive, and the Schwartz lab measured basically how much the cells could spread when he expressed either integrins, expressing alpha-2b beta-3 versus alpha-v beta-3. And he basically saw these interesting results, which is for relatively soft substrates of the order of 5 kilopascal Young modulus. Alpha-2b beta-3 seem to present enhanced spreading, which translates into higher propensity of adhesions to form, which further translates into faster activation of these specific integrin isoform with respect to the alpha-v beta-3. So in summary, we started from this question: how cell adhesions respond to the external rigidity of the substrate. And when we simulated adhesion assembly using Brownian dynamics and incorporating these different catch bonds to sense stiffness, we could demonstrate that changing the catch bond kinetics alone can be used to understand differences in adhesion assembly in response to external forces. And when we also simulated the extension and activation of individual proteins, individual integrins, we could find that different isoforms of integrin actually present differences in extension, which translates into easier activation versus harder activation under force. So how cell adhesions respond to external force? On one hand, they can modulate their catch bond kinetics by changing the lifetime of their bond in response to force. And at a smaller scale, they can basically respond to force by changing their extension and activation dynamics. And this is all I wanted to present today. I want to thank you for your attention and thank my lab members that are listed here. A lot of the experimental work that you guys saw in this presentation comes from experimental labs, obviously. We only do simulations and modeling, so I need to thank all of my collaborators, in particular Yu Feng, that did those single protein extension experiments, Tristan Driscoll and Martin Schwartz, as well as Patrick Oakes from Loyola. I would like to thank the funding agencies. We currently have fundings from NSF, NIH, and DOE, as well as some resources from the University of Utah Health here at the University of Utah, and the Center for High Performance Computing that allowed us to run the simulation at a high performance. I would be very happy to take any questions you might have now.
Thank you so much. That was awesome. There's several questions in chat. Can you see chat?
Oh, I do.
If you could read a question out loud before answering, that's going to go for the recording.
Okay. I see several of them. I'm trying to go to the first one. Has the system represented by the adhesion clash model been reconstructed in vitro with purified proteins? I think there has been a lot of tries. Lindsay Case at MIT is studying this. She's working with some theoreticians that are thinking of this assembly in terms of phase transition as opposed to modeling single particles and their assembly, but I know that she is exactly trying to reconstitute those adhesions assembly in vitro. Please indulge us with your educated guess. How far away are we from making a motility system reconstructed in a vesicle so we can have a synthetic cell motility? So my educated guess is that we are very far, but I don't think everybody would agree with me. I've never seen anything like this. If I think about a vesicle, I imagine the vesicle as a system where people have studied lipid composition and deformations. I'm not aware of people studying motility there. Do you want it to be, oh, somebody else have answered this question.
Or can I
Please.
follow up on that question? Yeah. Yeah, so I wanted to make the distinction between genetically encoded like motility systems versus just motility systems in general. So for non-physiological, non-genetically encodable motility systems, do you have an idea of when that could appear? Because that would be easier, I think. But yeah, I want to get your opinion.
I have never thought about that. So my very naive opinion is that this would happen in 20 years. But I'm not familiar with the field enough to have a really educated guess, I would say. What do you think? I mean, you guys that are like into, I'm assuming that since you are asking the question, you might have some knowledge and more than me in this field. And what's your opinion on that? Or somebody's opinion on that? Are we
I'm
close?
not sure.
Are we close? Like, is there any instance where those systems move to begin with? Or are they static?
The closest I've seen is people in microfluidic channels have been able to get actin assembly, like an actin comet can move around in a microfluidic chamber. And then maybe you can put that in a liposome and maybe that can move, but I haven't seen anyone try
Doing
anything
that.
else.
Yeah, there must be
Yeah.
obviously some.
But it would have to be like very simple, like it's like a rotation of a liposome or not even like linear movement. Yeah.
Oh, I see. So it's gonna represent some displacement, but not the mesenchymal or motion or amoeboid motion of cells directly. That would be a step. That would be a first step. You're making me curious. I should read something in this sense.
And last thing, I wanted to ask, who was the professor you said that worked on reconstituted systems that was trying to reconstitute the stuff in vitro? I didn't write that her name.
Oh yeah, Casey, Lindsay Casey, Casey. She used to work with Claire Waterman at NIH, and I think that's where she started, like studying mechanosensing of adhesions, adhesion assembly, and then she moved for a postdoc to another lab. Now she's a faculty assistant professor at MIT, and she's following exactly this question, how adhesion can be reconstituted in vitro. And she uses, different conditions and tries different ways to see these, what she calls phase transition of components into adhesion assembly. Do people investigate cell adhesion being influenced by environmental factors like pH, salinity, H? They do. I'm aware, obviously, of those studies where they change ion concentrations and induce the activation. For complex self-assembly, like ribosome assembly, what are some modeling strategy you would try? The time scale for ribosome assembly in vivo is about 2 minutes. A lot of the assembly is mechanical, but there are also many chemical modifications that need to be studied. So with a time scale of the order of minutes, you would try something closer to Brownian dynamics. It depends on the number of particles that you have in the system, but if you start with very simple first order kinetic reactions, you can capture 5-10 minutes of dynamics. Thank you for the talk. What simulation software do you use? How large of intracellular systems are you able to simulate, and how much computational resources is required for the larger simulations you run. So for the Brownian dynamic simulation, we write our algorithm. I used to write code in MATLAB, Java a long time ago, and now my postdoc is writing Python code for that. We disseminate the code on GitHub. So all of our software is open source for use by others. And for the atomistic simulations, we are using GROMACS, which is a very popular software allowing for simulation of proteins. You can use the CHARM force field, which is very well validated for simulations of protein and ligand binding interactions. We have GPU nodes right now. We have 4 of them. For a simulation of integrin of 20 nanoseconds on 4 GPU nodes, the time that we need is of the order of a day or 24 hours, 30 hours of simulations. Have you also looked at or interested in gene regulation response to the mechanical signals you're modeling? We have not. We are focusing entirely on protein structure, function, and macromolecular assembly at the moment. We haven't gone to gene regulation. Other professors working on self-assembly modeling and in vitro reconstitution. David Oddy is working on adhesions and adhesion assembly. Somebody from Johns Hopkins, Sean Sun. Mofrad from Berkeley, Mohammed Mofrad, he's working on adhesions, response to mechanical forces. You're asking me who are my competitors.
Colleagues, more likely.
Colleagues.
I just want people to talk to or ask questions.
Absolutely. Yeah. Those are the first 3 that come to my mind. I have worked a little bit with Roger Kam during my PhD. He's at MIT in the Department of Biological Engineering. I'm not sure he's still working on that. I'm not sure he's still looking at adhesions, but he's a mechanical engineer himself, and he has looked at adhesions and all atom simulations and mechanosensing. The 3 people I have on the top of my mind right now are those 3, Sean Sun, David Oddy, and Mohammed Mofrad.
And how do you spell Oddy for David Oddy?
ODDE.
ODD.
Thank you. He's your neighbor. He's at the University of Minnesota. He's a good friend of mine. We published a review together on adhesion mechanosensing, and he came out with the motor clutch model, published in Science some years ago now. And now he has made a lot of progress on this model. So he started from a little spring, beads and spring system, and now he's able to simulate the whole cell movement in space, depending on stiffness and other stimuli. He showed that there is an optimal stiffness for cell migration velocity, which is very interesting because he detected a very new stiffness threshold or optimum, which happened to be not too stiff and not too soft. So the cells don't only move fast on stiffer substrates, but they have an optimum. And this optimum can vary depending on the cell type. So by incorporating the motor clutch model into a bigger model that simulates cell migration, it could characterize velocities as a function of stiffness. And I think this is one of the most recent work that he has done. But he's very interested in the interplay between individual adhesions and cell movement on matrices or substrates with different mechanical properties. And then Sean Sun does more atomistic simulations and coarse graining, whereas Mohammed Mofrad is focused on other components of the cell, but he has some paper about tallinn and vinculin, so adhesion proteins deformation under force as well. Cool, thank you so much for all of those questions, including those a little outside my field regarding gene regulations and vesicles, but it's good food for thoughts. Thank you so much.
It definitely is. Thank you so much. Thanks for the great discussion. And I hope you have a great rest of your Monday.
Thank you. Thank you. You guys,
See you, everyone.
have a good week. Bye-bye.
Bye.